Welcome everyone to the Cytognos workshop for the ICCS 2020 virtual meeting. My name is Irene González. I have a dual role in sales and support. I am the key account manager for the East Coast in Canada, and also the site and support manager at Cytognos USA. Today, I'm presenting Infinicyt Software and its FDA 21 Part 11 compliance tools. So this is our agenda for today. I will start introducing Cytognos, who we are, what we do, and where we come from. Then I will move to our next generation flow solutions that include Infinicyt Software. And we'll focus on the new security and logging module to support the 21 CFR Part 11 rule. Finally, we will jump into the software for a live practical analysis. So let's start. Cytognos is a clinical flow cytometry company that offers end-to-end solutions for oncohematology, clinical immunology, and clinical trials. Our aim then is to enhance patient care and improve the efficiency and workflow in the lab through our standardized, efficient, reproducible, and highly sensitive solutions. Here's a little bit about our history through the years. We were created back in 1986, so we have been around for about 25 years. And we were initially founded within the hematology department of the University of Salamanca in Spain. In 2006, we started to collaborate with the Euroflow group in bringing its validated flow cytometry method to the market, including the selected antibody panels and the Infinicyt analysis software. And from there, we started to commercialize the lyophilized reagent kits. We developed an automated analysis approach, and we incorporated a CE-IVD instrument, the Omnicyt. So as you can see, we have grown over the years, expanding our portfolio to offer a complete solution, and also expanding the applications of our solutions for clinical labs. In 2019, we opened our first office in the US, based out of Boston in Massachusetts. And the most recent update is that we have included the 21 CFR Part 11 compliance tools in the software. Cytognos is a global company. We are present in over 54 countries in the world across the 5 continents. Our headquarters lie in Salamanca, Spain, but we have offices in Boston to cover the US and Canada. So we may start by asking a question. Why does the quality of our clinical data matter? The clinical lab plays an essential role in the healthcare system as it provides the information that enables doctors to make the right diagnostic and therapeutic decision for their patient. So we as clinical laboratory professionals have a direct impact on many aspects of patient care, including in making a direct improvement in the life of the patients. Now, how can we improve the quality and consistency of the data that we generate in our clinical flow cytometry lab? Although the requirements might be different depending on the type of study, for example, if we are running a screening assay or a minimal residual disease test, there are some common points like using validated antibody panels, following standardized instrument settings and laboratory SOPs, using a data analysis approach that reduces the user bias, and also having software tools that ensure the traceability and security of your patient data. Our next generation flow solutions, which are based on the Euroflow developments, address all these challenges, as they include the validated antibody reagents, the standardized SOPs, the Omnicyt flow cytometry instrument, and the Infinicyt software. Plus, we offer the technical support and training required to implement these solutions. Now let's talk about Infinicyt Software. It can be used for any type of flow study, and it has interesting features to help you with multicolor data analysis, like the file merge, to combine all tubes from a panel and analyze them together. The population visibility, so you can specifically look at your target population by turning off the background. The reference image, or setting a population as a reference for following up patients. And the validation diagram, to study cell validation in one single plot. Additionally, we have advanced tools like the multidimensional analysis to separate different populations using all the parameters. We also have the clustering tool to eliminate the bias of manual gating. And our unique automated analysis and reporting approach using a reference database. I invite you all to watch our another workshop at the ICCS that is dedicated to this automated analysis and reporting for myeloma MRD. And to enhance data security and traceability, we have a complete security and logging module, and this is what we are going to see today. But if you want to know more about the capabilities of Infinicyt, feel free to contact us. We will be happy to walk you through all the features. Now, this new module includes 4 main features, which are the user access control, the audit trail, the password protected files, and the electronic signatures. All these together can help you meeting regulatory compliance and standard requirements of your clinical lab. So let's begin with the user access control. We can assign roles with specific permissions for each user in the lab. For example, we can define a technicians or analyst role with limited access to the software functions, and the pathologist or lab supervisor role with no restrictions. The roles and permissions can be customized, so we can choose the permissions that are going to make up a particular group or role. In this example of analysis workflow, we have the technician that opens the case, loads the profile, loads the analysis strategy, and certifies the analysis. Then he or she saves the analyzed files and exports the report. Next, the pathologist opens the analyzed file, reviews the analysis, makes changes if needed, and approves the report. The pathologist, in this case, will also define the analysis templates and the gating strategies. Again, this is just an example of a specific analysis workflow. You can customize your roles and permissions anytime so they fit your needs. Then we have the audit trail. Infinicyt records every action that is performed within a particular file. It will record the action, who did that action, and when and who did the signing. Then you can pull out all this data into a spreadsheet, and this is very useful for audits or lab inspections. The third security tool included in Infinicyt is the ability to protect your analyzed file with a password. This way you can keep your patient data safe and secure and protect your confidential information. So for example, when you have a challenging case and you need a second opinion from a colleague, you can safely send the patient data out of your hospital network. Or if you are in a multi-site clinical trial where the analysis is centralized and you need to transfer all data to one particular site. Finally, you can electronically sign your analysis and report before sending out your results. In Infinicyt, we have different signature status. We have analyzed, approved, and issued to indicate different parts of the analysis workflow. So in this example, the technician can first certify the analysis, then label the report as analyzed. Next, the pathologist can sign off the case and label the report as approved. And finally, label the report as issued when the pathologist is ready to send out the results to the clinicians. So now that we have explained these 4 options included in the security and login module, let's jump into the software to see them live. Okay, here we have Infinicyt Software. First, we are going to log in as admin. And the admin is able to access the account control, put in the users, and define which permissions make up a particular role. So in this account control window, we have the users list, the list of all the software users in our lab. We have the admin as the default user. And also we have Maria. Maria is a technician. We have Hannah. Hannah is our pathologist. We have John. He's another tech. And we have Robert, who is our lab supervisor. The software comes with the complete and basic group or role. And we have set 3 additional roles in our lab: the pathologist role, the technician role, and the lab supervisor role. And each one has different permissions. So the pathologists have no restriction. The technician can do the analysis but cannot make modifications on the analysis template. And the lab supervisor can edit everything but cannot sign off cases. So this is the particular setup that we have for our lab. But we can define the level of access for each role. And here we can see all the options that we have available in Infinicyt. So we have all the users in the system, we have defined the roles, so we are ready to start analyzing cases. We're going to log out as admin, and Maria is going to log in. Maria is our lab technician. And she has an LST case to analyze, so she's going to open the file in Infinicyt. This is the default workspace. So before starting, we are going to switch to our specific analysis template or profile for the LST panel. So Maria is going to load the profile. Now we have the right template for the analysis. Okay, we are logged in as Maria. So I'm not able to perform some actions. For example, I cannot edit the profile, I cannot save the profile, and I can't not edit the population tree either. These options are all grayed out for me. But what Maria can do is load in the analysis strategy. So the LST is a screening tube for chronic disorders, and we can identify the major lymphocyte populations and its subsets with this tube. And this is basically what our gating strategy includes. So let's review the gates now. Let's start with the debris doublets. We have one gate for the debris, and it looks okay. And we have another gate for doublets, but we need to adjust this gate because it's capturing singlets here. Okay, then we have our lymphocytes. We have 2 gates now, one for the forward inside the scatter, another one for the 45, and they all look good. Now let's check the T cells, the CD3 positive cells. The gate looks good, so let's move to the subsets of the T cells, the CD4 positive cells, the CD8 positive cells, and the double negative or DIM. Alright, then we have our B cell compartment. As you can see, we have to move this gate to be able to capture the CD19 and CD20 positive cells. And within the B cell compartment, we have our kappa and our lambda cells. Finally, we have here the NK cells. Okay, so Maria is done with the analysis. This is a normal peripheral blood case. And now she's going to certify the analysis. And for that, she needs to enter the password again. Okay, now the analysis is certified. And she can open the report. So in this analysis report, we have the patient information on top, we have the statistics, and some diagrams showing the pattern of expression of our populations. Now we just need to label the report as analyzed. As you can see, the approved and issued options are not available, as Maria is a technician. So we clicked on analyzed and enter our password again. Now our report is labeled as analyzed. And also we want to include our electronic signature on the report. We want to sign it as analyzed. So let's do it. Okay, here we go. Maria has completed the analysis now, and she can forward the case to the pathologist for review. So let's save the analyzed file. And that's it. Okay, now Maria is going to log out, and now it's time for Hannah, our pathologist, to go over the case. So Hannah logs in Infinicyt. Alright, and she selects the case that Maria has previously analyzed. Okay, now that I'm logged in as Hannah, and Hannah is the pathologist, I have the ability to make changes on the report and make changes on the population tree if I need to. Okay, what Hannah is going to do now is opening the report and review the populations. Remember that this report has been previously analyzed by Maria and labeled as analyzed as well. And now Hannah, after reviewing the statistics and the populations, she's ready to approve the report. And when she's ready to send out the results to the clinicians, she can label it as issued as well. Finally, we want to add the electronic signature of Hannah on this report. So let's add it. First we have the approved signature, and then we have the issued signature. Okay. Now that we are done, we can export this analysis report as a PDF, or we can transfer it directly to our LIS system. And obviously we need to resave this analyze file. Okay, so this case was easy, right? This was a normal sample. But imagine you get a challenging case and you want to share it with your colleague Elizabeth to have a second opinion. And Elizabeth is a HIMPATH that works in a different institution. So before passing the case along to her, we want to remove all the sensitive information and also protect the file with a password, so only her will be able to open it. So we want to resave this case with a different name and mark the anonymous option to remove the sensitive information from the patient, and mark the request password to open the zip file as well. So now we just need to type in a password and we will be ready to share this file with Elizabeth. Okay, so when Elizabeth gets the case, she just needs to log in with her username and password. So, okay, and open, she just needs to open the LST case that Hannah sent to her. Now she needs to enter the password that was provided by Hannah if she wants to open the file. And here we go. We have the file opened. Okay. And the last feature that I wanted to show you today is the audit trail. So let's say 3 months later, you have an audit in your lab and the lab inspector requests information on who analyzed the case, when, and who did the signing. So Robert, the lab supervisor, needs to pull out all this information from the file. So for that, Robert logs in. She opens the specific analyze file he wants to check. In this case, it's going to be the LST case that Maria analyzed and Hannah signed off. And he just needs to open the actions log. The actions log captures the information for each action performed within this particular file. We have the action, we have the type of action, we have the user, the date and time, and some details here. If you don't need everything, we can filter the information by type of action, we can filter the information by user, and also by day or by time. And from here, all this information can be easily copied and pasted into a spreadsheet. So the information is available for the lab inspector. Okay, so now let's go back to finish the presentation. So to wrap up, with Infinicyt Software you can integrate all of your files and analyze them together. You can do a multidimensional analysis using all the parameters. You can do an automated analysis and reporting using a reference database. You can ensure the quality and safety of your clinical data with the 21 CFR Part 11 compliance support, and much more. So here's our contact information. If you have any question, you want more information about Infinicyt, or you just want to know what Cytognos can offer to your lab, please feel free to reach out to us. Okay, I'm going to end here. Thank you so much for watching this video, and take care.